Today, the first at-home brain stimulation device just got FDA approval for major depression, and the pivotal trial data is finally peer-reviewed. A patient tried one antidepressant. It was not enough. The dose is right, the adherence is right, the diagnosis is right, and the depression is still there. Until recently, the next step in most US clinics meant either switching medications, adding another medication, or sending the patient to a clinic for transcranial magnetic stimulation, which often means weeks of daily in-person visits. But in January 2026, the FDA approved something different. A prescription brain stimulation device a patient can use at home, 20 minutes a day at the kitchen table under physician oversight. This is ProlivRx, and the data behind it is finally peer-reviewed. ProlivRx is developed by Neurolief, an Israeli medical device company, and it just received FDA Premarket Approval, the strictest device pathway, the one that requires direct clinical evidence of safety and effectiveness. The device is a wearable headset. It delivers focal, multi-channel stimulation to 2 sets of peripheral nerves, branches of the trigeminal nerve across the forehead and face, and the occipital nerves at the back of the head and upper neck. The stimulation is gentle. Patients describe it as a mild tingling, and each session runs about 20 minutes. The treatment is prescription-only, physician-directed, and is indicated for use at home or in clinic. The mechanism has a name, eCOT-AS, or external combined occipital and trigeminal afferent stimulation. So why these 2 nerves? The trigeminal nerve, cranial nerve 5, is the largest cranial nerve. Its branches fan across the face and forehead. The occipital nerves supply the back of the scalp and upper neck. Both nerves send sensory signals up into the brainstem. From the brainstem, those signals project into higher brain regions involved in mood regulation, autonomic function, and affective processing. This is a peripheral to central route, a way of reaching mood-relevant circuits through the body's own sensory wiring, rather than stimulating the cortex directly. The target is not a single brain region. The trigeminal and occipital afferents converge on brainstem nuclei that connect to the locus coeruleus, the dorsal raphe, the thalamus, and limbic structures. These circuits are the same ones that conventional antidepressants, SSRIs, SNRIs, atypicals act on indirectly by changing serotonin, norepinephrine, and dopamine tone. eCOT-AS is doing something mechanistically different. It is feeding structured electrical input into those same networks through the sensory gateway, and asking whether that input can shift circuit activity in a clinically meaningful way. The exact downstream network effects are still being mapped, and the FDA approved the device on clinical outcomes, not on a complete mechanistic story. Pivotal evidence comes from the MOOD study, a randomized, double-blind, sham-controlled multicenter trial. 124 adults with moderate to very severe depression, a Hamilton Depression Rating Scale score of at least 20 at baseline, were randomized to active or sham eCOT-AS for 8 weeks. The trial was self-administered at home. The primary endpoint was change in the 17-item Hamilton Depression Rating Scale at week 8. After the double-blind phase, everyone was offered 8 more weeks of open-label active stimulation. Here is what the trial showed. At week 8, the active group had a mean HDRS reduction of 8.62 points, compared with 6.01 for sham. That difference was statistically significant, with a p-value of 0.0196. Remission rates, defined as an HDRS of 7 or less, were 21.3% for active treatment versus 6.0% for sham, p equals 0.027. The sham arm also improved substantially, which is common in depression trials. But active eCOT-AS produced significantly more improvement and significantly more remissions than sham. In the open-label extension, by week 16, the mean reduction approached 10 points, and 32% of participants met remission criteria. The treatment was well tolerated. 3 participants in the active arm withdrew for headache or migraine. 2 withdrew for worsening depression. No one withdrew for elevated suicide risk. So where does ProlivRx fit? The FDA indication is adjunctive after at least 1 prior antidepressant failure. That is a lower bar than the typical 4-failure threshold used for TMS coverage, and it is broader than the classic treatment-resistant depression definition, which usually requires 2 adequate failed trials. ProlivRx is not a replacement for first-line antidepressants. It is an add-on prescribed alongside ongoing medication. Compared with TMS, it is a different mechanism and a different care delivery model, home-based rather than clinic-based. Compared with ECT, it is non-invasive and does not require anesthesia. Compared with ketamine and esketamine, it does not produce dissociation, and it does not require in-clinic observation. None of these treatments are interchangeable. They target different patient populations with different logistics and different evidence bases. Few honest caveats. First, the MOOD trial ran 16 weeks. We do not yet know how durable the response is past 4 months, and we do not know how patients do on the device for a year. Second, real-world adherence is always a question with home-based therapies. Daily 20-minute sessions require motivation and routine. Third, payer coverage has not yet been defined. TMS took years to get broad coverage. It is reasonable to expect a similar trajectory for ProlivRx. And fourth, the device is approved as adjunctive, not as monotherapy. It is meant to be added to, not replace, ongoing antidepressant care. The Class III Premarket Approval is a real milestone. The next chapter is real-world implementation. Questions or episode ideas? Drop them below and subscribe for more psychopharmacology breakdowns. Educational purposes only, not a substitute for clinical judgment.